Welcome, I am Garima Tripathi from RKGIT Ghaziabad. Today we are going to discuss the topic spectroscopic techniques in phytochemistry, NMR mass spectroscopy. Spectroscopy is the isolated and purified plant constituents should be identified and its chemical nature should be determined. The plant compounds could be identified by their spectral characteristics. Spectroscopy is the use of adsorption, emission, scattering of electromagnetic radiation by atoms or molecules or atomic molecular ions to qualitatively or quantitatively study the atoms or molecules to study the physical process of a compound. The theory behind the spectroscopy is interaction of radiation. So, here as the science or the technique is utilized, the principle of absorption, emission of scattering. I must like to mention in that absorption says that the light which is entering through the sample has certain energy level, and once it enters to the sample, leaving the sample, some amount of energy is absorbed for leaving behind a thin trail. In case of emission, the amount of energy with which the light is entering into the sample would start emitting a little higher energy level of the light. In case of scattering, there are two incidences where the under these electromagnetic radiation entering through the sample would emit either outward scattering of the light or inward scattering of the light. So, this is inward scattering effect and this is outward scattering effect, and this scattering takes place owing to the nature of the atom which is present in the sample or the molecule, their ions or any other physical process that is involved there. So, this particular technique is utilized both to identify to analyze qualitative and quantitative parameters of the analytes. It can cause either redirection of these radiations or transition between the energy levels of the atoms or molecules. This transition could be either absorption, emission or scattering. Among the number of spectroscopic methods, first one is ultraviolet visible absorption, abbreviated as UV vis. Second one infrared or IR. The third one is NMR, that is nuclear magnetic resonance. The fourth one is the mass spectroscopy or the MS, and they play very crucial role in identifying the plant compounds. The NMR is a theoretically complex, but powerful tool for providing the information about the structure of a molecule in a given solution. So, that suggests that the analyte is to be dissolved in the given solution before exposing it to the magnetic resonance, which this particular electromagnetic resonance radiation will impact the energy levels of the nuclei, and various changes could be registered, and the compilation of all the complex changes are recorded, and then the structure is computed. Here we are having two types of NMR spectroscopy. One is the proton NMR and the other one is the carbon NMR. The proton NMR is sometimes abbreviated as H1 Nmr spectroscopy. It provides a means of determining the structure of an organic compound by measuring the magnetic moments of its hydrogen atom. In any phytoconstituent, the hydrogen ions are directly associated either to the carbon, and their interaction or a number of protons present, their interaction, the neighboring atoms present will affect the different electromagnetic radiation, and hence the structure can be computed. Theoretically, subatomic particles like electron, protons and neutrons can be imaged by spinning on their axis. In many atoms like C12 carbon, these spins are paired against each other. What I mean to say is, in these each electron is paired in case of these C12 type of atoms, but there and these those nuclei of atoms have no overall spin. That means, if there is pairing, that means two electrons or protons or proper pairing is there. In that case, there is no spinning of the nuclei on exposure to the electromagnetic radiation. But in case of deuterium, that is H1 or 13C, that is carbon 13, the nucleus does not possess an overall spin. The sample of the substances is placed in a solution of in an inert solvent between the poles of powerful magnet. There are two poles of powerful magnet, and in between the sample is kept. Protons undergo different chemical shift according to their molecular environment within the molecule. In nuclear magnetic resonance technique, we are trying to see the chemical shifts that on exposure to this electromagnetic in presence of two magnets, the type of chemical shift will allow us to determine the molecular environment. It requires some 10 to 50 mg of sample, and this sample could be recovered. The advantage of using NMR as a technique, spectroscopic technique, is that this is easily recoverable as compared to other techniques like mass, where the destruction of the sample takes place. The spin of the hydrogen atom can be promoted from lower to higher level by interacting with absorbing energy from electromagnetic radiation in radio frequency region of the spectrum. So, let us have a look again, that the spin of hydrogen atom can be promoted from lower to higher level by interacting with the absorbing energy from electromagnetic radiation. That says that once the electromagnetic radiation, the sample comes in the field of electromagnetic radiation, the absorption of energy takes place and causing the elevation of lower hydrogen energy state to a higher level of state, and results in spinning of these hydrogen atom. Separation between the two spin energy levels is a sensitive function of molecular environment of hydrogen atom in the molecule. Thus, hydrogen atom in different environments absorb photons of different energies. Since the molecular environment of each proton, each hydrogen atom is different in different molecules, the absorption of photons would of different energies will be there. The NMR spectrum of protons in a molecule is obtained by plotting the amount of energy absorbed by the spinning nuclei versus the frequency of radiation applied or RF, frequency of radiation or RF applied to the molecule. The spectrum provides information about the chemical environment of the spinning protons and can be used to deduce the atomic band patterns in the molecule. Spin between two groups of protons result in small interaction with each other and is called as spin-spin coupling, where the space between each coupling is called as the coupling constant. So, J is this coupling constant is denoted by J. Multiplication of the coupling and the magnitude of the coupling constant in deuterium NMR or HNMR gives information about the number of hydrogen atoms minded. It gives the information about the number of hydrogen atoms on the nearest carbon atom of the relevant peak, whatever peak has been achieved, seen in that peak the number of atoms associated with that particular carbon, such information is revealed and their chemical nature. But the proton NMR cannot give any information about the nature of the carbon skeleton of the molecule. So, in that case we use 13C NMR that could help us to solve this gap. Even in 13C NMR, the theory is same, the spinning, the exposure to the radiation, absorption of the energy level and the amount of radio frequency produced to the molecules, the difference between them will help us to compute the carbon skeleton and their nature. The theory is completely same for both the techniques. Both the techniques is decoupled and coupled spectra are recorded. In case we are to elucidate or deduce the chemical structure, coupled spectra is also recorded and decoupled spectra of each of 13C NMR and proton NMR is recorded. But the coupled spectrum of NMR is slightly different from that of proton NMR and is called as off resonating proton decoupling. 13C NMR helps to identify the number of unique carbon atoms in a molecule, the number of hydrogen atoms attached to the carbon atom, the environment of carbon atom and CC skeleton of the molecule. So, these are the three major informations revealed after 13C NMR. The modern NMR techniques are also available presently. So, they are hetero-multiple bond correlation NMR, that is HMBC. Then we have correlated spectroscopy, also termed as COSY, COSY. Then we have heteronuclear single quantum coherence, that is HSQC. Nuclear over Hauser enhancement spectroscopy, that is NOESY. Total correlated spectroscopy, also called as TOCSY. They have made the characterization of the natural products very easy. This is the schematic diagram or representation of the nuclear magnetic resonator. Here you can see the two poles of the electromagnets are there. In between, the sample in a solution is kept. these are the coils around the sample, which record the whose signals are detected and amplified, and finally a record chart is taken. Here is the coil which generates the radio frequency, and then around the sample in presence of these two magnetic poles. So, this is another illustration, the look alike of an liquid or HR mass is something like this. Here inside there are electromagnets, sample are put from the above, and this is how an NMR looks like. So, the peaks which we record are some representation like this. These peaks you can find, with every peak you will find some singlet, quadret, doublet type of representation. See here, every peak will have some one big peak, and then with this the deduction is done. Then they are major application of this nuclear magnetic resonance in the field of characterization of marker, quantitative NMR profiling, NMR fingerprinting. All these lead in the study of metabolomics and biomarker discovery. So, NMR plays a very important role in metabolomics, the study of metabolism related study, and the biomarker discovery. So, here comes the most important phytochemical research parameter, which is utilized during the drug discovery research in a phytochemical research, as well as in case of quality control, quantification and other sources, other parameters. Okay. Measurement of isotopic enrichment, then we have structural identification of the compounds of interest and the targeted quantification. All these again come under the category of biomarker identification, phytochemistry, characterization of a metabolic pathway, as well as mass spectrometry. The sample in gas and liquid or solid state is introduced to the spectrometer, followed by ionization. In mass spectroscopy, this is another sophisticated device where the sample has to be analyzed before the analysis or before the deduction of the chemical structure. On ionization and they are broken into various ionic fragments. Each fragment will reach to the detector in a different pace, will have a different journey, and their charge to mass ratio and the peak obtained will help us. We get the charge to mass ratio of each of the fragment, and that will help us to deduce the complete chemical structure. This technique has developed immensely and has contributed lot in the new drug delivery, drug formulation, new drug discovery, and in the pharmaceutical sciences. So, here the ion detection or data analysis takes place. We could get the exact molecular weights of the compounds in microgram amount of the substance as of the sample as well. Even in forensic labs and all, if the toxicity of certain analysis is to be done and the derived isolate comes at a very small microgram level also, that could easily deduce, we are capable of deducing the chemical structure of that isolate. Here volatilization of the sample, liquid or solid state is done. Whether the sample is available in a liquid or a solid state, we can volatilize in the mass, that is prior to the ionization. So, the first step in mass spectroscopy is to volatilize the sample. Then the second step is to ionize the volatile sample along with any technique, any preferable technique. So, there are various ionization techniques available. Commonly used one is the chemical ionization, then we have electron impact EI ionization, desorption ionization. Once the ionization proceeds, then we are having charged molecules present in a gaseous phase, because we have already volatilized, that means in the gaseous phase. These charged molecules produced by fast atom bombardment, also called as FAB, plasma desorption, that is PD, and thermospray and particle beam. So, next is the ion and its fragments are accelerated by the electrical or magnetic fields, and the ions are separated on the basis of their charge by mass ratio and are detected. The data produced by the molecular mass measurements or the fragmentation data enables us to elucidate the possible chemical structure of the molecule. The use of hyphenated or coupled spectroscopy or coupled techniques are very much common nowadays. So, it is hyphenated or coupled with gas chromatography, liquid chromatography termed as LC-MS, capillary electrophoresis along with MS have made the identification of natural product very easy. So, here is the schematic representation of the mass spectroscopy. You can see here the sample, there is a heating filament present, there is a ionizing electron beams are sent here. This causes the charging of the ionization of the molecule. The bombardment is also done, so that heating is done and bombardment or any other technique is used to produce these ions. Then in presence of electronic beam, they get charged and then they enter this circular path. So, this circular path, the large curved magnet is present, and following this complete path, according to their charge and mass in the electromagnetic electromagnetic fields, each components get separated out. Finally, they reach to the detector and their charge to mass ratio, the peak is recorded and the required compilations are done. The mass spectroscopy's most important application is to determine the molecular weight of the compound. A mass spectroscopy either alone or in combination with chromatographic separation is probably the most effective method of identification of drug constituents. It is applicable across diverse fields including forensic toxicology, metabolomics, proteomics, pharma or biopharma, and clinical research. Specific application of mass include drug testing, discovery, food contamination detection, pesticide residue analysis, isotope ratio determination, protein identification and carbon dating. See, isotope ratio determination is also of immense importance in phytochemistry, as here we are working for the natural molecules which should be bioactive in nature, and as already suggested, not all isotopes are biologically active, only some isotopes are one or two isotopes, sometimes other isotopes. So, identifying the biological active isotope is important, and the ratio of such active isotopes determination is important. Then pesticide residue in the control or in the standardization of herbal medicines or any other formulation that is intended to be used in food industry is also an important part of mass application. Okay. The role of mass in proteomics. So, proteo as the name suggests is protein, includes the characterization of protein and protein complexes. It includes the sequences of the peptides and identification of post translational modifications. Then comes the application of mass in metabolomics. So, in metabolic conditions where cancer screening in case of diagnosis, global metabolic fingerprinting analysis, biomarker discovery and profiling, biofuel generation, use and lipidomic studies, metabolic disorder profiling, all here the role of mass is immensely important. Then we have this application of MS in environmental analysis like drinking water testing, pesticide screening, quantization, soil contamination assessment, carbon dioxide and pollution monitoring and trace elemental analysis of heavy metal leaching. Next is the application of MS in pharmaceutical analysis. So, starting from drug discovery or new drug development and ADME studies that includes absorption, distribution, metabolism and elimination studies, pharmacokinetic and pharmacodynamic analysis, metabolite screening and preclinical development, MS plays the important role. Application of MS in forensic analysis, analysis of trace evidence with fibers in the carpet, polymers in paint, arson investigation example fire accelerant, confirmation of drug abuse, identification of explosive residues or bombing investigation to MS or mass spectroscopy gives its due. Now, another very important use of mass is its clinical application. During the drug development process, one is the regulation of the new drug, regulatory affairs of the new drug process. We need to take permission from the clinical drug development, the phase 0 studies or clinical test, disease screening, drug therapy monitoring, analysis of peptides used for diagnostic testing and identification of infectious agents for the target therapies, mass is applied. So, here are the references. Thank you.